name is Dr. Jennifer Fritz, and I'm going to talk to you today about DNA replication. All life on our planet is made up of cells, which are the smallest and most basic units that can perform all the activities required for life. Some of these activities include growth and reproduction. Molecules, such as proteins and nucleic acids, play an important structural and functional role in the cell and are critical for the continuation of life on our planet. Some life on our planet consists of single-celled organisms, whereas other life consists of multicellular organisms. In both cases, the cells that make up the organism must give rise to more cells as the organisms grow, divide, or reproduce. The continuity of life on our planet demands that all new life comes from pre-existing life, or in other words, all cells come from pre-existing cells. Over the past 3.5 billion years, life on our planet has expanded and diversified into the multitude of organisms we observe today. In our species alone, the number of cells found in one adult human body is about 50 trillion. A trillion is a million millions, or in other words, a lot of cells in just one human. If we multiply the number of cells in each human by the number of humans on the planet today, which is about 6 billion, we end up with a total number of human cells on our planet that is about equal to the total number of stars in the universe. How is this possible? How can one cell, the zygote, the product of fertilization, union of sperm and egg, ultimately give rise to the 50 trillion cells in the adult? By the process of mitosis, or cell division. In order for cells to give rise to new cells during the process of mitosis, many of the contents in the original cell must be doubled or copied prior to being distributed into each new cell. Think about this important concept. If the contents of the original cell were cut in half every time one cell divided and became two cells, the cellular contents would begin to approach zero after just a few rounds of cell division. Now, one important molecule common to all cells on our planet is DNA. DNA encodes information needed for the synthesis of RNAs, of which some RNAs are used in the synthesis of proteins. Complete and accurate transmission of DNA from one cell to two daughter cells allows each cell to have a full set of DNA instructions for synthesizing RNAs and proteins. Therefore, it's crucial that the quantity and the information in the DNA is doubled prior to being distributed during cell division. DNA replication is the process during which one entire molecule of DNA becomes two identical molecules of DNA that can be equally distributed into two identical cells. The structure of the DNA molecule, long sequences of complementary base pairs held together by hydrogen bonds, allows for complete and accurate replication of the original molecule. As a result, each new DNA molecule is composed of one original strand and one newly synthesized strand. This semi-conservative model of DNA replication is so accurate that only one mistake occurs every billion base pairs. These mistakes are known as mutations and are the ultimate source of differences in DNA between cells.